pleasure to introduce Dr. Patrick Keeling again for his second lecture in the Story Lecture Series. Dr. Keeling grew up in rural Ontario and only really got interested in science in his senior year of high school after a creationist was invited to speak in his biology class.
That was a mistake.
Rebelling against what he heard that day drove him into a degree in biology at the University of Western Ontario, a PhD at Dalhousie University under Professor Ford Doolittle, studying prokaryote-eukaryote transition. He was a visiting scientist in Melbourne, Australia with Jeff McFadden, a postdoc with Jeff Palmer at Indiana University. Started his own group at the University of British Columbia in the early 2000s, where he's become one of Canada's most prominent evolutionary biologists and a world leader in single-celled eukaryote biodiversity. Patrick's work has helped define the eukaryotic tree of life and has revolutionized our understanding of microbial symbiosis, endo and otherwise. He's a fellow of the Royal Society of Canada. He's a recent recipient of the Gilbert Morgan Smith Medal from the National Academy of Sciences. His talk today is entitled Comparing Parallel Evolutionary Transitions from Photosynthetic Algae to Intracellular Parasitism in Apicomplexans and Dinoflagellates. And if you ask me, we should send more creationists into high school.
It's backfired.
Is this working?
Yeah. Okay. They made that argument, actually. I had a letter to Science magazine about that said, see, we should have more creationists in high schools. I disagree. Okay, this sounds like it's backing up a little bit. Is that okay?
Yep.
All right, great. All right, need that. Okay, thanks. I changed my title because that was way too long. What happens to plastids that lose photosynthesis? And I'm just showing this because I love this picture and I want to know who put this up in the lobby because I think it's fantastic and I'm going to start using it in my teaching. So whoever you were, thank you very much. They protists, that's great. Okay, so in my lab, we are interested in the biodiversity of eukaryotes generally. You want to fix something?
I'm just going to lower them.
Is it on me?
No.
Okay. So this is a tree of eukaryotes and little thumbnails of some of the fantastic morphological diversity that we see mostly in single-celled eukaryotes. And just to orient you, here's the things that almost everybody in the world studies, which are the fungi, the animals, and the plants. And everything else on here we just sort of call a protist, although there is no real one definition of protist, but they're mostly microbial, but not all microbial. They're just basically eukaryotes that aren't animals, plants, and fungi. I like animals, plants, and fungi too, but not very many people like protists, and I go for the underdogs, so that's what I mostly work on. And when I say I work on protists, I mean I work on a lot of different protists, because this is kind of a fun field where you can dabble around and be completely superficial, and everybody thinks you're really smart. And I can be the most superficial guy in the Royal Society, so great. Because every one of these groups is something we've dabbled with, and the other problem, the flip side of that, of course, is sometimes you can't do anything but dabble, because we don't have all the tools, we don't have all the cell biology methods and stuff like that. So there's fun times and there's sad times when you get to the end of the road with some project and you have to just move on. I'm also interested in symbiosis and how it's impacted this tree and how eukaryotes originated. And that's mostly what I'm going to talk about today. And I'm going to actually focus in on a single one of these groups. And I'm going to sort of just kind of give you the idea of how fractal-like this is, because when you zoom in on any one of these branches, there's a ton of diversity inside it, too. Just like there's a ton of diversity, bilaterians. A lot of people would say they're very diverse. Sure. So are every one of these groups. So this is a subset of this group, the alveolates, and we blow that up. We have two major groups and a couple smaller groups at the bottom. The Apicomplexans and the Dinoflagellates are fairly well-known things, and I'm going to talk about those. These are really interesting. They have a photosynthetic ancestor. The ancestor is a mixotroph. It had a photosynthetic plastid, but it also ate stuff. And that's a really interesting setup to have for ecology and evolution. And there's a lot of really interesting character evolution. Both these groups have gotten very diverse and very interesting. And I'm going to talk about how this plastid adapted and changed as these things diversified in a couple different stories. Because one of the things that's interesting about mixotrophs is they can lose photosynthesis fairly easily. It's very difficult to lose photosynthesis if you're an autotroph and you don't have any other way to get food, but if you're already eating, you can lose photosynthesis and keep your plastid fairly easily. And these are all dots that just represent times that photosynthesis was lost. And it's actually just kind of made up here. I just started throwing dots on the tree because there's all sorts of occasions when photosynthesis has been lost. But it's interesting because on this side of the tree, over here, most things aren't photosynthetic, but there's been only a few losses, but they were old, ancient losses. Whereas on this side of the tree, most things are photosynthetic, or at least half. But there's been many, many, many losses of photosynthesis that are all more rare, more recent. So there's one big loss of photosynthesis here, changed a whole group. But these losses are all independent events. And I'm going to pick a couple of these and just dig into them because they're interesting. So first I'll deal with one over here on the Dinoflagellate side, and then I'll deal with the big one and talk about some of the differences that happen. All right, so Dinoflagellates, very, very diverse eukaryotes with lots of different, they can be parasites, phototrophs, heterotrophs, both, all three. And the student of mine, Liz, was really interested in digging into dinoflagellate genomics, but you can't grow most of them. And she was interested in heterotrophic dinoflagellates, and you really can't grow them. And when COVID hit, she stole this microscope and some other stuff and took it home and just started doing plankton toes off the dock near where she lived and going home and picking single cells and putting them into the buffer that we use for single cell transcriptomes. So the idea being you can sequence, on a good day, 10 or 20,000 genes from the one cell. But at the same time, you've taken a picture and a movie of that cell. So you have this visual representation of what it's doing, what it looks like. Then you have 10 or 20,000 genes from the same cell, which is a fantastic thing to be able to do when you're interested in diversity of things you can't grow. So these are the cells she picked during COVID, and she had them all in her freezer. And she, after COVID ended, well, whatever, didn't end. But after the labs opened again, she took them back and she made libraries out of every single one and sequenced a transcriptome of all these cells, which is just an amazing, like this, I don't know, quadrupled or more the diversity of dinoflagellate molecular data that we had available. And up till this, there was only maybe, I think, 2 dinoflagellates that weren't photosynthetic that had ever been sequenced, because most of the ones that are in culture are photosynthetic, and none of these are photosynthetic, or very few of these are photosynthetic. So she basically exploded the tree with data. Now, in this, and every one of these is actually pretty interesting, and I can't tell you about all of them because I only have a bit of time. So I'm going to package up one little group of interesting things. You see these guys with the little dot, like that pink one with the dot, and there's one beside it, and another one, and another one. Those little dots don't look like much, but those are really, really interesting little dots. And Liz became a great natural historian who said, oh, that's that dot. So it's one of those things that you learn somehow by osmosis. Because what those dots are is a tiny little camera eye inside the cell. So this is one of them. This thing's got a lot going on, okay? This is a crazy cell. So this is the dot. I'll get to that in a minute, because you're probably all looking at the piston. And the piston is this fantastic structure that shoots out many times the length of the cell. It's got this weird decoration all over it. Okay, we don't know anything about this thing. I can't talk about this thing because nobody knows what it does. Nobody knows what it's made of. Nobody knows how it works. Nobody knows what it's for. I've heard stories, they don't make sense. It's not, people said maybe it bounces around between sand grains, but this thing's planktonic. Nothing makes sense. The piston's a giant mystery. I would love to get some microscopy on the piston, but we don't have it. So I'm going to talk about the dot. There's some other stuff here you can't see, and I'll talk about it briefly too. All right, so here's what I want to talk. Well, actually, I'm going to go on a little excursion too, because this thing also has this, which is a nematocyst. It looks like the nematocyst from a jellyfish or from a coral. But it independently evolved this incredibly complex structure, which you can see blown up here from a fibsen. And it's basically like a Gatling gun-shaped toxic projectile that this thing uses to shoot out at its prey and paralyze them and then reel them in and then eat them. And then remember, this is all inside a cell, though. It's all subcellular. There's no tissues. There's no other cells here. This is a subcellular thing, but of amazing complexity. And the piston, I don't have anything to say. This thing is what I want to talk about, and this is called an ocelloid. Basically, it's a camera eye. And so this has been observed a long time ago, and people have speculated a lot about, wow, wow, this thing can see images and all this kind of stuff. It can't see images. It doesn't have a nervous system. I had a prof that showed me this in third year. I told a couple people this because it's kind of funny that I've started working on this. And I remember he got all philosophical, and he was like going like, at the screen and trying to impress us all with microbiology being cool. And he said, who sees what this eye regards? I thought, I'm hooked. That's great. Because it's a good question, because it probably doesn't see images. Of course it doesn't see images. Our eyes don't see images. Our brains make images. But it's probably a light harvesting thing that maybe increases the signal-to-noise ratio and maybe gives you some directionality or something like that. And it helps these things hunt their prey, which are dinoflagellates. How's it made, though? How did it evolve? We did FibSem on it, and we determined that the retina, the cup probably of light harvesting part, is actually a plastid, a non-photosynthetic plastid. And it looks like this. It's fantastically ordered structure with these crazy curved, very, very, very regular folded membranes. And somehow or other this absorbs light, probably. We don't know, but probably. And somehow or other that connects with something in the cell that tells it to swim somewhere, probably. That's how these things tend to work, so whatever. People argued a lot about how it works, but proteorhodopsin was the candidate that was pushed, because proteorhodopsin does this. It absorbs light, pumps a proton or some other ion, and it's used all the time in different protist groups for for sensory reasons. But proteorhodopsin's not in plastids. And so, but still, proteorhodopsin was pushed. So how do you do this, though? Liz did this for 2 years, and she found 14 of these cells. So how do you work on function with 14 cells that you can't culture? That's a tricky one. So what she did is these single-cell transcriptomes. And the power of this is that she found 14 of them, and she did them all. So we have repetition. And she also didn't just find one. There's 4 known genera of this, and she found all 4 plus another one that no one knew existed, and so she described a 5th genus. And she got genome-level data from all of them, from single cells. So these are the cells, the exact cells that correspond to the cells in the tree, because we go 1-to-1 correlation between a cell and a thing on the tree. And this is a tree based on hundreds of proteins, phylogenomic tree with very high resolution, tons and tons of data, okay? And what she found, here's the 5 genera. This is the new one, Cyclopsia. And we got a tree. So that's nice. So now we can put characters on this tree and study how they evolved. So we're already doing way better. So here's the tree again. These are the photosynthetic ones. This one's interesting because it's actually partitioned photosynthesis into part of the plastid, and then part of the plastid is the retinal body that's not photosynthetic. So it's got 2 zones of its plastid that are not or yes photosynthetic. So that's very cool. Warnawea was thought to maybe be photosynthetic because it's kind of green, but nobody really knew. And so here we go. Let's look at the photosystems. So this is what you'd expect. These are all photosynthetic dinoflagellates, and you get lots of photosystems. If there's a white box, it's usually because it's not a very good transcriptome, or we just didn't capture that gene. So these are transcriptomes. So we don't have every single gene. But you can see tendencies, like we tend to have all the photosystems, or like representatives of all the photosystems in the photosynthetic things, and that's fine. And in our 2 probably photosynthetic, Warnaweads, these are called, we also found all the photosystems, more or less. Not every gene of every system, but we found every system. This is a non-photosynthetic dinoflagellate, and this is what you expect from a non-photosynthetic dinoflagellate, is no photosystems, right? So that's this is what you expect. And you see this in lots of non-photosynthetic dinoflagellates. So what about our non-photosynthetic guys with an eyeball? Because that has that cool plastid. Well, they don't have none of the photosystems. They have some of the photosystems, but they've lost others. And it's a very specific pattern. They've lost photosystem 2. We'll go back to that. And these guys up here have also lost the ATPase. Photosynthesis was lost twice, once here, slight difference in how, okay? But they've both lost photosystem 2, and they've both lost everything to do with carbon fixation. They've lost rubisco, but they've kept the light harvesting complexes. So they're absorbing photons, but only in photosystem 1. Does anybody recognize this pattern? Because this is a physiological pattern that plants do But it's not a pattern that anything has ever evolved permanently. It's just what plants do when they're light stressed, because it's cyclic electron transport flow. So when plants get light stressed, they can downregulate photosystem 2, and that's the dangerous one that's breaking water and making oxygen, and also lots of ROS and stuff like that. So this is a very dangerous thing to have, and you got to control it. Photosystem 1's a little bit less dangerous. And so what they do is, rather than linear electron flow, they cycle the electrons back through B6F. And they can make a little bit of reducing potential and a little bit ATP. But they also pump protons. And so what we think's going on here is that we kind of had the obvious right under our nose. What could possibly absorb a photon of light and pump a proton in a plastid, right? Oh, it must be proteorhodopsin, which has never been observed in a plastid. No, it's a photosystem. They're already there. And we didn't find proteorhodopsin in any of the non-photosynthetic ones. We only found it in the photosynthetic ones, and they're probably using it to swim towards the light. So in this case, you know, I actually had a really hard time convincing a lot of photosynthesis biologists of this, but what I think is happening here, I think the only explanation for this, is that the photosystems have been repurposed. They're no longer an energy generating system, they're a sensory system. So they already had a light harvesting proton pump sitting there working, and when this became a non-photosynthetic plastid instead of sensory organelle, it just used the pump that it already had. And instead of using the proton pumping for energy, it's using the proton pump to create a gradient, which is probably feeding into some sensory cascade that we obviously don't know yet. And there's way too many candidates to be sure of how that could possibly work at this point. But that seems to be what's happening here. So we have, just to reiterate, I believe this retinal body, plastid, and it makes sense. It just used the existing machinery to do what we expect a photosensory organelle to do. But it still is, nevertheless, I don't think this has ever been observed before. I don't think the photosystems have ever been repurposed for a sensory package like this. But when you stop and look at it, you kind of go like, okay. I mean, I don't have any problems with this. I think it works. How the ATPase has been lost in some and not others is an interesting question, and maybe I can get some input on that. It's kind of fun, though, that there's some variation here. But yeah, this is a fate of a plastid that's lost photosynthesis, but it's kept a lot of the machinery. Just used it for a different reason. So then I want to move over to a more dramatic case of loss of photosynthesis that's attracted a lot of attention. And the reason why is because the Apicomplexans are all obligate intercellular parasites. And some of them are very important medical parasites. Like, for instance, Plasmodium is the thing that causes malaria up here. And when the plastid was found in Plasmodium and Toxoplasma, again, it was one of those things where Ian Wilson was telling people this, and people were making fun of Ian Wilson for saying something so ridiculous, and he was totally right, because everybody thought it was the mitochondrion. But these things have a plastid. These are localizations of the plastid of the malaria parasite by Jeff McFadden. It's now very well studied, by far the best studied non-photosynthetic plastid, and one of the best studied plastids of all, simply because malaria is such an important parasite. As soon as it came out, resources poured into it, and lots of people have done some amazing cell biology on it, and there's fantastic genetic resources for this thing. So this is a very well studied non-photosynthetic plastid. But where did it come from? How did it evolve? Those are all interesting questions that took a long time to work out. And there was a couple of really big breakthroughs. The first was the discovery of some new organisms, and that always shakes things up. These are photosynthetic organisms that were found associated with coral that people assumed were dinoflagellate coral symbionts, but they actually turn out to be related to Apicomplexans and not to dinoflagellates. So Vitrella and Chromera were fantastically important for understanding how this non-photosynthetic organelle evolved. My lab got involved when we sequenced the plastid genomes to address the question of whether this thing was a red alga or a green alga, and some debates about that, and that was great. We ended the debate. It's a red alga. This is what finally showed that the ancestor definitely had a plastid, and they didn't get two plastids independently, and that's great. But like other great projects, it also raised some new questions, because the student that did this, Jan, came back to me a little bit later and he said, hey, man, look at this. Some other people had already found Vitrella before anyone found Vitrella. And what he meant was that microbial ecologists found a quote-unquote new phylum of bacteria the year before Vitrella was discovered that was somehow related to cyanobacteria. They found it on a coral reef. And so they didn't know what to make of it, and this happens quite a lot. There's lots of new phyla of bacteria in microbial ecology. Having sequenced the plastid genome, we realized this isn't a new phylum of bacteria at all. It's a chloroplast of a eukaryote that's just a bit weird, and so they don't have too many chloroplasts in this tree, and it didn't branch with the chloroplast, it just was outside of them. And so then well, how many more of these are there? How many chloroplasts were sampled by bacterial ecologists that didn't have a very good alignment and called it a new phyla of bacteria? So I had a couple students go through this. We redid it when the database grew so fast. Basically what we found is from hundreds of millions of sequences from lots of environments, there's hundreds of thousands of misidentified plastids in the bacterial database. And almost all of them fell within an algal group. In other words, there weren't a lot of new kinds of algae that we'd never heard of before. There's actually a couple that are coming out now where people have done this with bigger databases and found really rare things, but we didn't find it, except for a few unknowns. Not a few, but we did find a few, and they were all related to Apicomplexans. They weren't in the apes, but they were outside the apes. And they were all from coral reefs. And this isn't trivial because it's about equivalent to 10,000 species. So there's 10,000 species of new things out there that all have chloroplasts, and they're all related to Apicomplexans, and they all live on coral reefs. That's what the data say, from millions of reefs. So how do you find them? Okay. All right. This is just to show you where they go in the tree. And Vitrella is related to one of these groups, Chimera is related to another, but there's like up to 15 of them now, 15 different groups that are almost all associated with coral. There's a couple of exceptions, but they're almost all associated with coral. So we want to find them. We picked this one because it's the most common and the closest to the apes, and so we decided to go out and get it. So we got to look at coral. This is what took me to coral reef research. Happily, it's been great. We want to know, is it in coral? Is it on coral? Is it a photosynthetic algae or is it a parasite? You can't tell just from the ribosomal RNA. Does it cause disease? We don't know any of this. It's just a sequence, right? But we know it's abundant. It's all over coral reefs. That's nowhere else. So I started this out I went big. I invited all my friends. And we had this huge expedition that went out to Curacao every year with about 30 people where everybody came and brought students and we all kind of worked together, and it was the best meeting I've ever been to because there were no talks. And I made very good friends with Forrest Rower and Mark Vermey, who runs the station. I brought a cat home once. It's been really pretty awesome for me personally. I've been there lots of times now for over 8-9 years. And it's become a really big part of the research program, and I'm so happy I did this. And it worked, too. So we had a big breakthrough a few years ago. Walden Kwong, postdoc in the lab, found these things. We call them Corallicolids now. This is Arrow 5. We call them Corallicolids. He took pictures of them. They look like Apicomplexans. They're not green. They're not photosynthetic. They're parasites. He found what tissue they're in. They're in the mesenterial filaments, in the periphery of the mesenterial filaments. And he sequenced the plastid genome. So great, we're really making progress with this. We still don't have a nuclear genome though, and I'll come back to that. Because the plastid genome set us off on another tangent. Because it showed definitively that they're parasitic, they're not photosynthetic. Well, they're symbiotic without being photosynthetic, so we call them parasites. There's no photosystems. They can't be photosynthetic. But they do have chlorophyll biosynthesis. And this, take my word for it if you don't know this already, is really weird. You shouldn't have chlorophyll if you don't have photosystems. It's like having the bomb without the bomb squad. And these are only 4 genes in chlorophyll biosynthesis because these are the only 4 that should be on the plastid genome. Most of it's in the nucleus. These are the only 4 that the ancestor would have had. These are the only 4 that Vitrella has, et cetera, et cetera. So, but nevertheless, this is striking. What on earth is this thing doing with chlorophyll? Not only that, the plastid and nuclear data we had were conflicting on where it went in the tree. So remember, the plastid data put them way down deep below the Apicomplexans, down here, right? So Corallicola is early. Well, we got a couple nuclear genes from a really bad transcriptome with a busco of 0. And one of them was the ribosomal RNA, and it put Corallicola's way up late in the tree, near the coccidia. And so neither of these is conclusive, and we need more data. So we had to get a genome or a transcriptome or something from these things. So, took us back to the tropics. This is Curacao again, one of our sampling sites, and we had to work out model systems. We had to work out protocols for purifying or whatever, because you can't grow them. So a lot of time at the microscope. We identified 2 hosts that are 100% infected, that are both really easy to collect, and they're not endangered or anything. They're weedy things. And we developed Percol gradients that will enrich massively for the parasite compared to Symbiodinium or whatever. And we could get from that EM the first live pictures of these things. We could watch them doing their gliding motility. We recognized what they look like. We got good at picking cells. We got all these resources. This took years. All these resources nailed down. But it was really hard to get a transcriptome. Finally, this student here, Victoria, broke that problem and got beautiful transcriptomes from both hosts, the Madrasis and the Parazoanthus. And I say beautiful, this is how good the data set is, this bar. And you can see the Corallicolids are about as good as the genome projects from the medically important parasites. And this is from natural samples that we cannot grow. So I'm actually quite proud of that. And this is a phylogenomic tree, 201 genes, a lot of data. And it definitively shows that Corallicolids branch up here late, near the coccidians. So the plastid gene was wrong. This is correct. They're actually up here. So, just zoomed into that. So, going back to this little schematic, this is wrong, this is right. Okay, so what does that tell us, though, about the character evolution of the chlorophyll, right? Because actually, if they branched down here, it was fairly easy to explain, because they have chlorophyll and the other Apicomplexans don't. So you could say, well, photosynthesis was lost down here and chlorophyll was lost 1 branch later. It's weird, but it's not super weird. But where they go in the tree means that chlorophyll had to have been maintained all the way up through the backbone of Apicomplexan evolution and then lost repeatedly over and over again in every Apicomplexan lineage except for this one, which is really weird. And a lot of people jumped on this and said, oh, it's something to do with living in coral, but it also can't be that because all of these ancestors didn't live in coral, probably. And so there's something else going on. Lots of parallelism, though. And that's, I don't mind that because parallelism is all over this tree. Apicomplexans have parallel evolution of just about everything. These guys down here were actually mistaken for a particular genus of Apicomplexans because they have the exact same form of parasitism, and yet it turns out they're not even branching in the group because they evolved parasitism in parallel. We've lost photosynthesis lots of times, and I think we've also just lost chlorophyll a whole bunch of times. So this group here has chlorophyll, and these groups here all had to lose it for this tree to be, for this character evolution on this tree to make sense. But like I say, parallelism, we have to get this through our heads. These really complex things are evolving in parallel over and over and over again, and you can't look at this with parsimony and make sense of it because it doesn't work. Okay? All right. But this also begs the question, does that really make chlorophyll? Really? Because look where it branches in the tree. It makes no sense. And I said there's only 4 genes on the plastid genome that are involved in chlorophyll biosynthesis. The rest of them are encoded in the nucleus, and they're targeted to the plastid. So we didn't have the nuclear data, so we couldn't really say they were there. And that includes the definitive CHLG enzyme that is the one that makes it into chlorophyll. It's a very important missing piece of the puzzle. And so we go back to the transcriptome data from these 2 hosts, and now we can say, yep, they make chlorophyll because they have every one of these genes in the nucleus. They're all expressed, and they're all under purifying selection. So they're actually making chlorophyll. These are still functional genes that the organism needs. So that's kind of nice. I mean, it's nice, but it doesn't tell us why. And here, I honestly just don't have an answer. I have some speculations, and they're kind of, I go to the boring. So here's a just so story. For example, I don't believe this is true, but I think it might turn out to be something like this. If you had an alga and it was using chlorophyll excess to downregulate heme biosynthesis on a whole, if it was a feedback inhibition system, and then you became non-photosynthetic, you might need to still make a little bit of chlorophyll in order to regulate your heme biosynthesis, even because you need to keep making heme. Everything needs to make heme. And so I was wondering if it may be something boring like that. And now we're working out ways that we can sort of test that kind of a model and see whether it makes sense. But again, remember, these are non-cultivatable parasites that only grow inside the tissue of, zoanthids that are symbiotic on sponges that live under 20 meters in the Caribbean. So it's not a great model system, but nevertheless, this is a very important question, because I think this is the only organism on Earth that we know makes chlorophyll but doesn't do photosynthesis and have photosystems. I think that's true. I haven't found another one yet. So if you know otherwise, let me know. Okay, and I want to end. I still have a few minutes, right?
Yeah.
Yeah, good. I want to end with a good news COVID story. We all love those, right? Because COVID completely messed with all this. This was a brutal. We had trips crashing and burning in 2020, and that's why this took so long. But it made us think about things differently. And how do you carry on doing interesting work like this in British Columbia? So when you think about sampling in the ocean, you tend to think, I like this guy, You think about really butch men deploying shit from their ship, and but basically what they're doing is they're sampling the water column a lot, and occasionally they go down and get some sediment, because that's really hard to do. And that's great, but it's missing stuff that's in animals, say, because they're filtered out. And so imagine if you went to Africa and you said, I want to know about the distribution of malaria, so I'm going to sample a lot of air, and I'm going to filter out the mosquitoes and sequence for malaria. You would come up with very little malaria, and you'd wonder, well, what's up with all these sick people? Are they just kidding around? Like, what's going on? It wouldn't work. And some parasites broadcast, and some don't. Some of them have very tight, controlled life cycles through different animals, and they do not broadcast into the environment. So this is an example of one that broadcasts. Malves are the most abundant sequences of eukaryotes in the ocean. And we've been working on these too, because it's kind of fun. These guys have lost their plastid entirely. It's one of the few groups that have done that. And we recently found out that these malves aren't even a group. They actually evolved twice, because now we have free-living relatives, the alephrids and the oxyrus, that are each related to different malves. And so we can say the most abundant parasites in the ocean are actually not a group. They're two different groups that evolved twice independently, so more parallelisms. And their plastids evolved differently. These guys didn't lose their plastid, and these ones did. A few weeks ago, we all assumed they did too, but they're not even related, so they didn't. So that's just a little neat story about a broadcast parasite that you can get tons and tons of for the ocean. But what if the Corallicolids don't do that? Well, one outshot of that is that maybe they don't just live in coral reefs, because we don't do that kind of sampling anywhere else, because people love coral, they sample coral, but we don't sample other kinds of animals related to coral in other places. So where I live, it's a pretty nice place. There's a lot of ocean, there's a lot of biodiversity in the ocean, including things related to coral. So I pitched this project during COVID to a whole bunch of postdocs and students, and they all said, nah. And this undergrad, she was like, yeah, I'll do that. That sounds great, because she'd been stuck during COVID, and she had this research project in the summer, which is mostly on the computer, and at the end of the summer, we're like, do you want to go out in the ocean and look for parasites? And she's like, sign me up. And she ran with this. So, yeah, British Columbia, we've got corals, right? They just don't build reefs. They're little cup corals. We've got anemones in spades. We've got more anemones than coral reefs do. We've got loads of anemones. We have zoanthids, like our model system. Parazoanthus is a zoanthid. We have corallomorphs, which is our other model system that Walden Kwong first worked on. These were misidentified as, they're called strawberry anemones, but they're actually corallomorphs. We got all this stuff in British Columbia. Octocorals, I hate octocorals though, because they're so hard to collect. But we do have, sea pens are octocorals, so we have them too. So we went out and sampled a lot of this kind of stuff, and it turns out there's loads of Corallicolids on the dock right in front of a cabin I go to in Galliano Island off the coast of Vancouver, where UBC is. And so this is the early data, but basically this is a tree of Corallicolids, and everything with red is from a coral from warm water, and everything in blue is from an anemone from British Columbia. So there's tons of diversity of these coral parasites. They're just not turning up in people's sampling because of the way we sample. They're anthozoan parasites that are globally distributed. They're everywhere. And so you might also notice a pattern here. All the blue ones go together and the red ones mostly go together. And it begs a few questions. Is there cold clade, because it's cold water? Or is there an anemone clade, because these are anemones? And these are corals, and these are hot. And so Morelia went a little further and got full-length ribosomal RNAs from a whole bunch of our star candidates. One is Metridium. This is a white anemone that you get here. It's very, very common here. And from the Corallimorph, which I assume you must get here because I think its species name is Californicus or something. So I think it's also down here. These things are 100% infected. We can visualize the parasite with fish in their mesenterial filaments. We know it's in the same tissue. But we also went back to the Caribbean to get anemones because we want to test the warm-cold, anemone coral thing. So you can test it both ways. This is harder because you have to go out at night because most of the anemones are small and they're transparent. And so she worked out that if you use the fluorescence, you could actually zero in on the anemones really quickly. And if you're fast, you can get them out of the sand before they retract and dig themselves down. So this is one of the ones that we've been collecting. And this is actually a Corallimorph that I just put up because I love the picture. I love taking fluorescence pictures underwater. It's like crazy beautiful anthozoans. But we've also collected them too. And if you do that, so this is a tree of full-length parasite ribosomal RNAs. And these columns have different we're looking for a pattern. So in the first column, we have the host with different kinds of anthozoans. And there's no pattern. It's just kind of a mess, right? The second column is the temperature, cold and hot. And again, it's kind of a pattern, but not really. The third one is biogeography, and I think that's actually the answer, is that there's two clades, one of which is almost entirely Atlantic, and the other one is entirely Pacific. And so it looks to me, we got to do some more work here, but it looks to me like there's biogeography, but there's not much host specificity. I think these things can be generalists, and I think they definitely jump around from different host to host within the Anthozoa, even in British Columbia. But there does seem to be some real big differences between the Atlantic and the Pacific varieties. So yeah, this is great too because it gives us fantastic travel opportunities, because we really want to now go look at things from the North Atlantic. There's tons and tons of anemones in the North Atlantic and nobody's looked in them. We need to go to the South Pacific, of course, right? But yeah, we need to get some more genomics because we've got at least two big clades, and the two things that we have got transcriptomes from are agonizingly closely related to each other, so we'd really like to get some more diversity. And we really need to figure out how to dig into this question about chlorophyll, because that's the tough one. That's the most important one, but also the way hardest question here. So I got a nice opportunity in Vienna this summer to gather together a whole bunch of people that used to be in my lab together with my lab and have this nice picture, so I'm going to make everyone look at it. But mostly I want to thank, Victoria did like massive amount of work to get that genome data from the Corallicolids, and Morelia's whipped up this cold water stuff and the anemones, and they've both been a real pleasure to work with. And a number of other people that did a bunch of the deep background and worked on the Percol gradients, and Sam Livingston's helping out with the imaging. So, these things are teed up for FibSem, and we're going to try to get 3D structures and some detail from them. We've got lots of plans, so that's fantastic. And also, particularly the Gordon and Betty Moore Foundation, who really, I probably supported every single thing I talked about, except for the Tula Foundation, who supported the work on the Dinoflagellate single-cell genomics at the start, and Liz Cooney's work through the Hackeye Institute. I'm very grateful to both of those. Thank you.
Questions?
Sounds like, yeah. What?
No.
No,
Yeah.
but this was on, remember?
I have to turn mine off?
Yeah.
Okay.
Oh, not the front one. Turn off the front one, not the back one.
This is live. Top.
I think it's the other way around. There, go. That should be on now.
Okay, yeah. Can you take the cells from the Percol gradient and just hit them with a violet laser pointer or stick them on a transilluminator and see chlorophyll?
We have done that, and you can't. So they're not pigmented. There's no autofluorescence. I'm actually not familiar with. Do you need them associated with a chlorophyll binding protein to get the autofluorescence, or does chlorophyll do autofluorescence just as a molecule?
It's
It
not,
needs.
I think, really fluorescent unbound. It generates tons of triplets, actually, is my understanding. But
Yeah, but we have hit Corallicolas with various kinds of lights, and they don't have any fluorescence.
So, just an announcement.
Yeah.
Hello? Oh, yeah. I'm also going to be monitoring the Q&A on Zoom for anyone who wants to post questions there. But I'll keep going in person.
Hi, Patrick. Amazing talk. Thanks very much. Since you're working with single cells, how much variation in the transcriptome is there between transcriptomes of a single type? And is that possible to affect some of the trees and things that you build based on single cells?
Sure. So, is this on now? Or I'll just check. So the question is whether single cells have enough variation that they might affect the tree. So almost everything I showed you is a single transcriptome from a single cell. The only exception is the Corallicolids, where we did a whole bunch of single cell transcriptomes, but then we also took Percol gradients and directly sequenced that, and then pulled all the Corallicolid stuff out just by virtue of its similarity to other Apicomplexan sequences. So we have a bit of more depth of data. And what we can say from them is that, so in the case of the Dinoflagellates, every one of those is its own cell, and they all branch exactly where they should branch, and there's almost no variation within something that you would call a species. So Dinoflagellates also have a lot of copies of most of their genes, and so you'd almost have as much trouble from that paralogy, like really shallow paralogy might cause problems too. But it's at the level where when you're reconstructing a phylogenomic tree of genera, it doesn't really come into play.
Just a little follow-up on that. So I assume you collected, you probably don't go collecting at night, and since these things have, some of them have light sensitive, is there
we do collect
genomic
that.
variation by,
Yeah.
Is there
Right.
chronobiology related?
Yeah.
We do collect at night.
You do collect.
Yeah, and the question, if you ever find the exact same thing at night and day, is tricky, don't know. If you find different transcription profiles, I don't actually believe that single cell transcriptomes are particularly good for things that are quantitative. I see them more as qualitative. We just get a lot of data and use it. But I wouldn't necessarily trust a single cell transcriptome from a dino at night and day and say, look at the different expression profiles, unless there is something just off the charts different and that you could get repetition. Like if you had multiple cells from the day and multiple cells from the night, you found the same thing. Yeah, maybe. But this is why,
Do we?
yeah, not in my lab, but somebody should. But this is why I focus on whole complexes. So I'm confident to say that photosystem 1 is there and photosystem 2 is gone, just because we did it over and over again with multiple cells from the same genus and found no evidence whatsoever for an entire complex. But absence is a tricky thing with transcriptomes.
So one more question, if you allow me to follow up with one more on that. So have there been no surprises
When genomes have been sequenced from some of the, say, the cultured single-cell eukaryotes and compared to transcriptomes where you thought things were lost or not, and the genome says something different from the transcriptome?
I wish. As far as I know, no one's ever sequenced a genome from something that we've done a single-cell transcriptome from. But we're going to test this, though, in a different Apicomplexan parasite because we're looking at this question of how they expand their genome and contract it during different life cycle stages. So we're going to start doing genome sequencing instead of transcriptome sequencing on one of the things that we have single-cell transcriptomes for. Yeah. Oh, and just to finish with the Corallicolids, the two different hosts, the ribosomal RNA was identical. And when we were bummed out, we thought, oh, it's the same parasite infects both of them. And when we got all this data we looked into this at a nucleotide level because we wanted to see whether we could tell the difference between a generalist that infected both versus two really closely related specialists. So when we got all the single cells and all the slurries, we looked at a nucleotide level at the variation within each and compared it between each. So it sort of answers your question. And the answer was within each was almost no variation, and between them was a few percent. And so we were convinced that there was two specialists. But it also gets at your question that there's almost no variation between different cells from the same host with different single cell transcriptomes.
With respect to the hypothesis that chlorophyll may be a way of pulling out heme and modulating the levels of heme, is there any evidence at all that intermediates between magnesium protoporphyrin 9 and chlorophyll are toxic to the cells?
I don't know of any, and actually I should say I don't have any evidence of anything with my scheme.
In any system.
Yeah,
I don't, not your systems because you can't manipulate it, but there must be mutants of the chlorophyll pathway.
I'm totally sure there must be, and I don't know about it, so I'd have to
That would
defer
support
to.
your idea, your hypothesis a lot more than just assuming it.
Yeah. So
Because to have the extra genes means you're making more proteins,
Yeah.
you're stressing the cell. You only have one protein that could pull it out.
So you're saying if you had a different, like an earlier intermediate that could feed back and kill why would you bother going all the way to chlorophyll?
Exactly.
Or the other way to look at it is that the system evolved to detect chlorophyll and it doesn't detect the other intermediate, so it's kind of locked into going all the way to chlorophyll. You know what I mean?
Yeah, I don't know how tight the pathway is controlled. There are feedback loops in it,
Yeah.
and there's oxygen requirements.
Yeah. There's a lot, I mean, I'm not convinced my idea is a good idea, it's just the only one I got.
That's a literature thing that you could deal with.
Yeah,
My
no, it's
second
good.
question was, referring to these as parasites.
Mm.
Parasite to me means it's along for the ride.
As
And
opposed to
there's
pathogen.
no requirement for a metabolic interaction between the two. Is that the way you're looking at it, Joe?
Well, I use the word parasite very sloppily, I admit to that. So it's a symbiont, for sure. Whether or not it's pathogenic, I'll get back to. A parasite implies that it's bad as opposed to a commensal, which a lot of Apicomplexans probably are commensal because they're just really good at not hurting their host. With the Corallicolids, I'm actually now pretty convinced, based on the work of another lab, that they are parasitic, because it turns out that if you're infected with a Corallicolid and you bleach, you die, and if you're not infected and you bleach the same host, they can survive. And so it's like it's kind of kicking them when they're down, I think. So it doesn't cause any obvious disease in the context of a healthy coral, but in a coral under stress, it's harmful. So I think we're okay calling them parasites, but they're sort of conditional parasites and probably commensals in the systems we're looking at. And I don't know about anemones, say. I should call them commensals. But a lot of people don't know what that is, so I just say parasite because I'm lazy. But you're absolutely correct.
Patrick, really enjoyed your talk. So
The process of purification of these organisms, particularly from the host, and you're obviously looking at an organism that's undergoing a life cycle. Do you see cell division, and how does it look like? Because I'm obsessed with cell division and microtubules, and I want to know, like, do you see them divide?
Okay. All right, this is a fabulous question. We have pictures of them inside host cells where they're in the parasitophorous vacuole with 4 cells, which is a real standard coccidian thing to do. So those are just recently divided. However, these guys have complicated life cycles. Coccidians and most of the Apicomplexans up in this part of the tree have complicated life cycles. And a lot of them have 2 hosts. And so something that we're growing with growing dread, I will say, that I think we've only seen half the life cycle, which means there's another host. And it's probably the host where they have sex, which is the definitive host. And so now we're spending a lot of time going like, ugh, like, how do you go out and find the animal that is interacting at such a close ecological way with the coral that it's exchanging corallicolid parasites without releasing them into the water column or the sediment, because you never see them there. So there's probably some, mollusk or something out there that's the definitive host of these things. And it also has to be something that's in British Columbia, and it's in Curacao, and it's in Japan. So it's a really common, mollusks, a lot of parasitology. I talk to my invertebrate parasitology friends, and they go, I wish it was a polychaete, but it's probably a mollusk, that kind of thing. And I'm like,
Could be in the stomach of the animal.
Yeah, yeah. So, I mean, if you look at how the well-known parasites in this part of the tree go, there's almost certainly going to be another host. And that really bothers me because it's going to be so hard to
I
find.
wonder if they change shape dramatically. That's the
Maybe.
transition their whole organelle setup to.
Could be, yeah. I mean,
a
that
lot of amoebas show up like, have no microtubules, for example, and have all of a sudden show centrosomes, microtubules divide, show flagella, and then disappear all of those things and become amoeboid again.
Yeah, and these things, luckily for us, there's tons of work on coccidia, and it's a good place to start. These are not coccidia, they're related to a thing called protococcidia, but it's not that far off. And so I have actually almost got to the point where I'm going to sort of say, you know what, I'm going to sit back and let someone else find that, because, and that's probably how it'll happen, and someone will say, hey, look what I found in this mollusk, and yeah, rather than going out and looking for it. Because I gave that a lot of thought, and that is actually really hard, because there's a lot of animals on a coral reef, and we don't want to collect every single one, and they might not be 100% infected, so you'd have to collect a lot of every one. And I mean, I like scuba diving, but not that much, so I don't know what to say. But thanks for ruining my day.
There's a couple questions from Zoom from Clark Gregarious. The first one is, can multiple corallicolids infect the same coral and possibly exchange plastids and or nuclear DNA?
Okay. I would say they might be able to. We don't have any evidence that different corallicolid strains have infected the same corals. We do have a little bit of evidence that they might be a little sloppy. They might be generalists that can move around between different species. And because they're all in the mesenterial filaments, and because the infection level is not usually too heavy, I couldn't rule out the possibility that different parts of different filaments in the same, or different, polyps in the same colony could be infected with different. And we don't actually work on stony corals much now that we've identified that they're everywhere, because stony corals are protected, and it's just better for everybody if we just focus on these soft-bodied things instead. So we don't work on these massive colonies much. I wouldn't rule that out as a possibility, but I would say I very much doubt that they'd ever exchange anything, because they'd have to get inside the same cell, because these are intracellular parasites, and what we see is them sort of scattered around the periphery of the mesentery, and we don't see them in huge clusters. And it's probably true that once you're infected, once the glidosome has pushed a parasite into an animal cell, that it's probably hard for another parasite to get in. So I really doubt that. But in the other host, they'd be having sex, and there what happens, who knows?
And the second question from Clark is, are any of the nuclear genes involved in chlorophyll biosynthesis, e.g. CHLH, CHLI, or CHLD, retained in corallicolids that have lost essential plastid genes involved in chlorophyll biosynthesis? So I guess genes remaining in the nuclear genome for chlorophyll biosynthesis, but lost from the plastid.
Okay, so this is a tricky problem because you'd have to have a lot more data. As far as we know, no. But first of all, remember these are transcriptomes, and so we have a bunch of transcriptomes from different individuals of 2 different species of parasite. So they might not all have all the genes and all the transcriptomes, and that could be sampling. The other problem is we actually don't have the plastid genome from exactly the same parasite that we have the transcriptomes for. So there's a hole we're trying to fill now, just to go back. But there is corallicolid plastid genomes from several corallicolids from both clades, the Pacific and the Atlantic, and they all have the same chlorophyll binding gene, or biosynthesis gene. So there's no reason to believe they don't all have the same 4 genes in their plastid genome. There's no reason to believe they don't have all the genes in the nucleus as well. There's only gaps that are probably sampling gaps from transcriptomics.
Another question in the room?
Okay, this is really wildly cool to me, but I was wondering specifically about the retinal bodies. If from the transcriptomes you can tell whether there's any significant deviation of these retinal body PS1s from the photosynthetic versions.
So at a sequence level, are the PS1 proteins in Warnowiids different from photosynthetic things? Not really. They're a little divergent, but they're not crazy. Like we actually have our eye out for crazy divergent things because we do that sometimes. These ones are a little odd, not too odd. It looks like they're fairly ordinary. So Nothing untoward, I wouldn't say. They seem fairly ordinary. Oh, sorry. I have to keep turning this off. So.
All right, yeah, I have a follow-up question. So in photosynthetic electron transport in chloroplasts, another complex is involved that connects the ferridoxidin produced by photosystem 1 to the quinone that is oxidized by the B6F complex. You don't show it in your schematic. But there's known as the NDH, or there's two different versions, actually, NDH and PGR5, I believe, in plants. Is there evidence that that complex is also present?
Oh, I should have written this down. Yes, but it's not the normal one, it's the weird one.
Okay, so the weird one would probably be the PGR5 one that doesn't conserve energy. So the NDH complex, which is the ferridoxin quinone oxide reductase, is proton pumping. So it
it's not that
energy.
Yeah,
right. The PGR5 one does the electron transfer, but no, it doesn't conserve energy.
Yes.
Which is interesting because that would produce less of a proton gradient for ATP synthesis. So it could be one of the reasons why ATP seems to be lost in some of the cases. Though you still get proton gradient production at the B6F. So there's some energy being conserved.
And the reason why this pattern jumped out at us was because shortly before this, we worked on a different system. Sorry, this is going to be a really long answer, but it's kind of cool. Which is a kleptoplast. I talked about it last night. It's this Ross C. Dinoflagellate that has a kleptoplastic haptophyte that's related to the tertiary haptophyte plastids and dinoflagellates. And when it steals the haptophyte plastid, it does cyclic electron transport instead of linear electron transport in its photosynthesis, even though the haptophyte did linear electron transport. And it uses the same complex that I can't remember the name of that you just mentioned, which is why we were looking for it in here. And there, this is interesting though, because this system arose by losing this. That system arose this way because to build this system from nuclear genes that you have to take from a haptophyte and target to your kleptoplast only required 3 genes. Whereas if you added photosystem 2, you need to get 10 more genes from your food that you can target to your kleptoplast. And so we think it's like a sort of like a poor dinoflagellate's kleptoplastic photosynthesis, where they haven't acquired enough genes to have the full-on photosynthetic system in that case. But they arose, they kind of came to the same place, one by building, one by degrading, if you follow me. So it was an interesting sort of, it's not really a parallelism, but, and they both use the same non-standard linker that you were talking about.
Absolutely amazing talk. Thank you very much.
Thanks.
Yeah, there's no more questions.
Thank you.
Thank you.